Cytoskeleton. This session is going to look at a principal component of cytoskeleton, that is microfilaments. Uh, Microfilaments are also more commonly called as actin filaments. And the moment you think about actin filaments, you basically think about actin myosin in the skeletal muscle and how they work in tandem to enable contraction and relaxation of the muscles. So therefore, microfilaments have an important role to play in not just giving a structural integrity to the cells, shape, etc., but it also helps in the motility of the cells per se. So let us look at the learning outcomes of this session. So microfilaments, a principal component of the cytoskeleton, are coiled coil structures, which means there are two polymers coiling around each other as right-handed helices to form what is called as the coiled coil structure, and they are flexible and strong. These are made up of actin molecules, which exist in two states, G-actin and F-actin, with the latter, which means the F-actin being the actin when it is part of the polymerized filament. Microfilaments have polarized structures showing what is called as dynamic instability. That is, one can observe that actin filaments are assembling and at the same time at some other place disassembling, or the same actin polymer can assemble and disassemble. These are able to resist compressive and tensive force. Um, Microfilaments are highly versatile, functioning in cell crawling, amoeboid movement, changes in cell shape, cell signaling, and muscle movement. Let us look at the structure of the microfilaments first. The word microfilaments comes from the fact that out of all the three cytoskeleton, actin filaments are the smallest in size at 8 nanometers. And the microfilaments are practically made up of actin, and actin can exist in two states. One is what we call as globular actin or the G-actin when it is not part of the filament as yet. So it has a very typical conformation, and then when it becomes part of the polymer, that is the actin filament itself, it exists in the state of F-actin where its conformation is different from that of the G-actin. Now, as you can see, G-actin is a single polymer, and this folds typically into forming different domains. So there are several subdomains, as you can see over here, subdomain 1, 2, 3, 4, and within this 4 subdomains is a region where it can bind to a nucleotide, and the nucleotide specifically is ATP. So every G-actin will be found bound to an ATP molecule. This is also an indication that G-actin has therefore ATPase activity, which means it just does not bind to ATP, but it can also hydrolyze ATP to ADP. And therefore, this functionality of it being able to hydrolyze ATP into ADP enables giving a particular kind of regulation to polymerization and depolymerization of the actin filaments per se. Now, when you look at the F-actin, okay, before we go into the F-actin, the G-actin present in the vertebrates, there are three isoforms of the G-actin, alpha, beta, and gamma, with alpha actin primarily found in muscle cells, while beta and gamma actin are isoforms that are found in non-muscle cells. The actin, of course, polymerizes head to tail and in a right-handed helix. So each actin polymer is winding around another actin polymer. So you can see in yellow it is one polymer, and what you see in brown is the other polymer. So both the polymers wind around each other, or what we call as coil around each other, and each of them has a right-handed helix structure. So the two right-handed helices come together to form a coiled structure. And with every G-actin now in the polymer, it is called as the F-actin, and every F-actin has an asymmetric structure with one side being pointed and the other side being barbed. So therefore, in a filament, that is actin filament, one end of the actin filament is always pointed and the other end of the actin filament is barbed. Also, what is interesting is that the ATP binding cleft always points towards the minus end. So this is how you will always have the actin present in a polymer with this facing the minus end. Here there is again the same kind of understanding as it is in microtubules, that the minus end of the actin filament is the slow growing end, while the plus end of the actin filament is a fast growing end. So you can already see that based on this structure, they are already polarized. So one end is pointed and the other end is barbed, but also it is polarized in terms of the fact that you have more of polymerization happening at the plus end and more and less of polymerization happening in the minus end. So this is a slow growing end compared to this one, which is a thoroughly fast growing end. Now, this is a typical electron microscope picture of an actin filament, and what you can very clearly see is these are the barbed ends. So it's like an arrow. When you have an arrow, one end of the arrow is pointed and the other end is barbed, and that's how the name has come plus. So the plus end is always a barbed end, and the pointed end is the minus end. So one can observe that when it is a free subunit, this is the conformation of the G-actin. But when it becomes part of the polymer and it becomes F-actin, you can immediately see how the conformation changes, such that one end is the barbed end and the other end is the pointed end. Now the plus end elongates 10 times faster when sufficient G-actin monomers are present in the system. So there are several experiments that were carried out, and so you can understand that the plus end is definitely the fast growing end. So an experiment when conducted in vitro with the solution having a salt and a certain salt concentration and of course the actin subunits, what was observed is that the actin subunits first come together, so the G-actin first could come together to form an oligomer, and it is this oligomer that becomes a nucleation a nucleation oligomer for further growth of the actin filament. So as you can see over here, during the lag phase, the nucleation takes place, and once the nucleation has taken place, then you can see how you have the actin filament growing. And this growth of the actin filament will continue as long as there is a higher concentration of monomers. Once the concentration of monomers is equivalent to the concentration of the F-actin, so when G-actin and F-actin are more or less in the same concentration, so they are basically at a steady state equilibrium, so you will find that at both ends of the actin filament, you have addition as well as you have removal of the actin happening. So this happens at both the ends. So what would happen is you have a constant equilibrium being maintained by assembly and disassembly of the actin filament, which is why it is said that the actin filament is always having what is always showing what is called as dynamic instability. That means it keeps assembling and disassembling, or you can use the word polymerizing and depolymerizing at both the ends if the actin filament is free, with of course the understanding and the observation that at the plus end the polymerization as well as depolymerization is relatively faster than polymerization and depolymerization at the minus end. Now the same experiment, when it was carried out in such a way that at the 0th time oligomer itself is added in vitro, then you would see that there is no lag phase. Why there is no lag phase? Because the oligomer is already nucleation center, and with the oligomer being added, you would immediately have it going into the log phase or the elongation or the growth phase, because the initial oligomerization time is not needed, since what you are adding is already a preformed oligomer, and that is why you will see that it will directly go into log phase. So the understanding of this experiment is that the formation of the oligomers has been observed with the actin filament, or what you call as a nucleation filament has been observed with respect to the actin filament. So this nucleation is important, where you have slowly and gradually a small filament being formed, and this filament then becomes the nucleation point for more G-actin to be added on both the sides, whether it is a minus end or whether it is the plus end. Now we go to what is called as actin binding proteins. So there are observed to be a lot of proteins that are able to bind specifically to the actin and thereby affect the polymerization and the depolymerization of the actin filaments, also enabling formation of different patterns or different arrangements of actin. And these different arrangements of actin enable different functions in the cell. So, say for example, you have profilin. Profilin is again an actin binding protein, and this profilin, on binding to actin, is actually promoting polymerization. So, you will see that there is enhanced polymerization when profilin is binding to actin, while if thymosin binds to actin, then there is lesser polymerization, or polymerization is inhibited. So in actuality, you would find that there is a competition between profilin and thymosin to lead to a higher polymerization or lesser polymerization of actin. So I'm just giving a few examples of actin binding proteins and how they help in the filament formation. Then you have what is called as ARP23 complex. This ARP23 complex, on binding to an activating factor, is able to act as a nucleation point for more actins to come and bind. So you would see that the activating factor and the ARP23 complex binds such that the minus end is bound to the complex while the plus end is growing. So in here, what you would observe is that there is a pause at the minus end, because you cannot have addition of the actin filaments at the minus end because of the presence of the ARP23 complex, but the ARP23 complex is the nucleation point for more of, or for the polymerization of the actin filament, with the actins being added only at the plus end. So this is something that one observes, that when you have one ARP23 complex with an actin filament growing, you can have another ARP23 binding to the same polymer, but then this becomes the base for beginning another actin, as you can see. And interestingly, what has been observed, that the binding of ARP23 to an existing polymer is such that it is going to result in an angle of 70 degrees between two polymers that are being formed. So you can see how a branch is being formed, and then eventually you will find that this will lead to such a kind of branch being formed. So the architecture of the actin that is formed is very different because of this ARP23 complex. So it gives rise to a tree-like web of actin filaments, and therefore this becomes an important architecture when, say for example, the membrane has to be supported or the membrane protrusion has to take place, then this kind of structures help. Now, say for example, you have formin dimer being bound to the plus end. Okay, then what happens is that due to the formin dimer being present, one finds that there is a capping happening at one of the ends, and the growth is happening only at the minus end. So there earlier, what we observed is that you have growth happening only at the plus end. Here, because of the presence of the formin dimer, you can see that you have the growth happening at the minus end. And what can also happen is that there can be a conformational change of the formin dimer happening to allow growth even at the plus end, but that depends on how the formin dimer is binding. So several capped population of filaments are possible where growth is present only at a particular end. So for example, end capping proteins such as CAP Z This capsid prevents the addition or loss of monomers at the filament end where the actin turnover is unfavorable. So wherever the capsid is present, there you will not have depolymerization nor will you have polymerization. And this can be observed in the skeletal muscle structure, wherein the actin at one end is always bound to a cap protein, and therefore at that end you don't have polymerization and depolymerization happening. Then say for example you have an actin filament bound to cophylin. So you can immediately see that when it is bound to a third protein, there is a change in its structure also. So you can have a cophylin coming and binding to make the structure a little different from the usual coil decoil structure, where you have now additional proteins coming and binding, and you can see how what is stretched into 74 nanometers now becomes contracted into 57 nanometers. So this is something that happens due to the binding of cophylline. Likewise, there are many actin binding proteins that can bind to actin and change its conformation, change its structure, change its architecture, and so on. Now, therefore, let us understand that suppose you have a fibroblast, okay, and within the fibroblast, as you can see, this fibroblast is giving rise to what is called as filipodia. In amoeba, what we know as pseudopodia. So you can see that how the membrane extends into what is called as podia, okay, and this extension is happening because the actin is growing. So, within this single cell itself, you can see how actin can be present in different forms. So, here, where the cell is taking the stress of the movement that is happening because of the filopodia formed. So, the filopodia is formed by binding to a substrate. So, you would have several surface proteins over here that can bind to a substratum, that can bind to an endothelium, that can bind to a particular support, and because of that what happens is that the foot can get a foothold, okay, and because it can get a foothold, there it is catching the foothold, and from here it is releasing the foothold. So where it is extending on this side, at this side it is retracting. So you have one retracting end and the other end which is extending. So you can see how the cell from this position will move further to this position. So you can see how the cell is crawling on a substratum, and so you can see that it is basically because of the arrangements of the actin within the cell that allows it to move in this particular function. So for example, where you have stress fibers, where you have the cell taking the stress because of the movement, there you can see how the arrangement of the actin is in parallel to each other, while here where it is acting as a shock absorber, you will find that it is present as a network, and so it becomes a gel like network. Because it is gel like, it is able to take, or it will able to kind of repress or kind of face compressive and tensive forces, where in these areas where you have to have slowly gradually a particular protrusion happening, you can see how you have the tree like arrangement of the actin happening, and where you have the filipodia formed, you can see how all the actin basically come together to form or push the membrane to form a filipodia. So within a single cell itself, you can see how the actin at different places is present in different structural arrangements to enable different functions, to enable forces to be balanced, etc. So therefore, this is a clear indication of the fact that you have the actin being continuously changing its arrangement, and that is happening because of dynamic instability. So this is basically trying to tell the same thing, that it can crawl on a substratum by flexing to form filopedia with a gel-like matrix just below the membrane to adjust to the changes in the shape of the cell. Now, we look at the molecular motors that are associated with these microfilaments. As you've seen that microfilaments are associated with movement. Therefore, for movement, some energy is required, and hence These actin filaments are associated with molecular motors, and immediately, instantly, what comes to mind is myosin. So there are 50 different myosins present in different cell types, which can come together to form myosin, and these myosins are the molecular motors for actin filaments. Now, just like dyneins and kinesins that are molecular motors for microtubules, myosins also are made up of several polymers, polypeptides. So you have two heavy polypeptides, kind of forming a coiled alpha helix structure with two globular heads, and it is this two globular heads that has the motor activity. That means these are the ones that has the ATPase activity, and at the neck and the hinge region are present several light chains. These light chains are the ones that get phosphorylated, and on getting phosphorylated, there is a change in conformation which activates the ATPase activity of the motor domain of the myosin. So effectively, it can bind to ATP. It can hydrolyze ATP and carry out motor activity. Interestingly, what has been observed is that many myosin come together. So you can see all the myosin tails come together in such a way that the myosin heads are jutted out, okay, while the tails form what is called as the core of the entire thick filament. This is something that you observe in sarcomere in the skeletal muscles as well, where you have the thick filaments. The thick filaments are the myosin, uh, all all myosin coming together to form the thick filament, and you have the thin filament, which is nothing but the actin. This is exactly what we are trying to look at. This is in entirety the thick filament, with one region where you have only the tail portion present, and hence this is not that thick, but the remaining part has the myosin heads as well. And you can see that this stretch of myosin has assembled with the tails, in this direction, while in this the tail is here and the myosin heads are here. So together, this set of myosin and this set of myosin is forming the thick filament. So these globular head domains of the myosin, they, as mentioned, are associated with the motor activity. They're able to bind to ATP, they're able to hydrolyze ATP to generate conformational changes, and this plays a very important role. Now, interestingly, what has been observed is that these myosin heads also have an actin binding site, so they can also bind not just to ATP, but they can also bind to actin. So we are now going to look at especially how myosin is able to bind to actin filament, and through its motor activity able to slide the filament or move the actin filament. So say this is the actin filament with its minus end and with the plus end over here, and this is the myosin which is bound to the actin. Now, interestingly, what has been observed is that in a muscle, when the myosin head is bound to ADP, not ATP, but ADP, then the conformation of the myosin head is such that it has a high affinity for actin, and it is bound in this form. So, if ATP is less, then generally you would always find that the myosin head is bound to the actin. This is also considered to be what is called as a rigor mortis. So when you know that you do not have ATP present in the body, then the muscles will become stiff because the myosin constantly keeps bound to the actin of the actin filament. Now, say for example, ATP comes and binds to the myosin head. As soon as ATP comes and binds to the myosin head, its affinity for the actin changes and it leaves the actin. So right now, the myosin head is not bound to the actin filament because the ATP has come and bound. Now, with the ATP being hydrolyzed, so when ATP comes and binds to the myosin head, the conformation of the myosin changes such that its ATPase activity is enhanced, and now you have ATP getting hydrolyzed to ADP and P. So with the ADP and P and the P still bound to myosin head, you can see how the conformation of the myosin changes, right? Where it was upright over here, now it has moved. In this conformation, with the PI being released, okay, and ADP bound, you can see how where it was earlier bound at this position, because of the change in the conformation, it is now bound to the actin present over here. Then when you have the ADP being released, you can see that this conformation again goes back to this conformation. So it is actually sliding the actin towards the minus end. So therefore, what the myosin is doing is that due to the power stroke, it is sliding the actin filament towards the minus end. Now you can imagine that when you have many myosin heads at the same time binding to actin filament, then the push that is happening or the sliding of the actin filament that is happening is going to be much more amplified when you look at only one compared to many. So when you have many myosin heads doing the same thing at the same time, then you would definitely have a bigger sliding movement of the actin filament, and that's how the muscle is going to contract. So let us look at that. So this is a thick filament, right? So this is one sarcomeric unit. You can see how the cap Z binds to the ends of the actin filament. So at this position, the actin neither polymerizes nor depolymerizes. It is only at this end that you have polymerization. Plus this is the minus end of the actin. So between one Z disc and the other Z disc, you have one sarcomeric unit. So when you have the power stroke happening, you will see that all the actin will bind to, all the myosin heads will bind to the actin, and the actin will be pushed in. So therefore, you will find that the movement of the actin, this actin is towards this actin, and therefore you would basically have the two Z discs coming closer to each other. So thereby the entire muscle is getting kind of contracted. And when you have the myosin leaving the actin filament, then again the Z disc will go to its original position and there will be relaxation. So this is how relaxation and contraction happens thanks to the actin myosin coordinated functioning. Then you can also have some myosins actually like the dyneins and kinesins walking on the actin filament. So as you can see over here, you have two motor heads of the myosin 5 which are basically swinging such a way that they are actually walking on the actin filaments, and as you can see it is carrying a mitochondria here, so it is able to carry out intracellular movement as well. So that is also something that is associated with actin and myosin. Intracellular movement is also possible because of that. Here also what is interesting, as you can observe, is because of the plus end is growing, you can have the protrusion happening and you can see budding happening in an yeast. So this is the mother cell, and because you have the actin growing over here, you can see that a bud is being formed. So you very well understand that as the actin grows over here, the protrusion keeps increasing and now you have a bud being formed. So let us make the conclusions. The microfilaments at 7 nanometers in diameter are filamentous and made up of actin molecules which have ATP binding site and myosin binding site, the myosin being its molecular motor. The available concentration of G-actin decides the polymerization and depolymerization at both the plus end and the minus end, with the plus end having a faster rate. Several actin binding proteins are able to influence the dynamics and the architecture of the microfilaments. The amoeboid movements of cells like the fibroblast, muscle movements, etc., are the manifestation of the actin myosin coupling and its regulation by other proteins. So the microfilaments or the active filaments with their different architecture are able to give shape and support, resist forces, extend membranes to extensions for movement, slide to contract and relax, and much more. Thank you.